Well, hello, everybody. How am I going to follow that?
Rockstar.
Rockstar. Right. Well, I thought I wouldn't give an ultra-straight talk, but I wasn't quite prepared to follow that. I was just interviewed. Try and explain to you what I'm going to do. I was just interviewed and asked, why did I become a neuroscientist? And I gave, unusually for me, an honest answer, which is that I really was interested in psychology and, more importantly, philosophy. And back before most of you were born, in the 1960s, I had the hunch, and I think it's probably going to prove to be correct, that we might be able to solve some of the important millennial-long problems in philosophy by studying the brain. In those days, there was really no way you could study the brain. There was no subject called neuroscience. You couldn't even take courses in it. But I thought that we probably were just at the dawn of the period in which that was going to happen. Now, as you know, all neuroscientists have enormous hubris. And we're not going to solve the problems of the philosophy and psychology. We're going to solve the economy. We're going to solve every possible problem that you can. Well, we actually can't. And I think, for example, the problem of consciousness is going to take many, 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 many more decades before we can even begin to get a grip on it. But one of the problems that I'm particularly interested in, have been interested in most of my life, is the problem of aesthetics, the problem of beauty. Why are some things beautiful and other things not? Why are some people beautiful? Why are some objects beautiful? And it's a really difficult problem in philosophy. And as far as I know, there really aren't any kind of mechanistic answers to it. So when I was invited to give this talk, I thought, okay, here's my chance. I'm going to take a flyer, and I'm going to abuse my position here and to speculate about how neuroscience would go about actually solving that problem. So that's what I'm going to do. And I think it's for you to decide whether I made a good fist of it or not. I'm used to starting off talks by giving a summary of what I'm talking about. And so here is actually what I'm going to talk about. There are 4 premises, and I'm going to try to address each one of them. The first is that the brain, I think we now all agree, the brain is modular. It's broken up into tiny little pieces. That wasn't always obvious, and for many, many years, even up into the 1950s, it wasn't clear that the brain could be broken up and studied as individually in terms of small modules. But I think that's now pretty well accepted. That's not to say there aren't processes which actually unite activity in lots of different parts of the brain, and that might be what consciousness is doing. But that's not something which is the main focus of a lot of people's research now. And these modules are organized, I think, in a quasi-hierarchical matter. That means you start at simple levels and you go to more and more complex processing as you go through the different modules. And I'll show you a little bit of evidence for that. And you finally combine into higher order features, which then eventually are available to parts of the brain which are involved in emotions, perceptions, memory, and things like that. So that's kind of just a background. I don't think this is terribly contentious, although people in the audience might have some qualms with it. Okay, now we get into the more interesting contentious parts. I think the aesthetic qualities of your experience, that is what you think is beautiful or not, might be part of a brain system for monitoring the goodness of activation of these different parts of the brain. There may be a higher order brain system which is actually monitoring the traffic going and the activity in lots of different parts of the brain and saying, yeah, that's actually activated this part of the brain maximally. And of course, what activates one part of the brain maximally is not going to be what activates in another part of the brain. So, for example, a beautiful face is one which maximally activates the areas of the brain which represents faces. And as I'll show you, there are areas of the brain which represent faces and objects and so on and so forth. So the idea is, it's just a way of monitoring whether you're doing a good job of activating that part of the brain. Things that activate it maximally are called beautiful, and so on and so forth. Now, here comes an even more speculative bit. One of the functions of the visual arts, and I'm talking mostly about painting here, is to provide stimuli and stimulation which selectively activate different brain modules. So the job of the artist is actually to come up with illustrations which are, originally they weren't terribly selective, but over the history of art, they've become more and more selective for activating different parts of the brain. And finally, if we look at my version of the history of art, it follows a trajectory which is actually the inverse of this hierarchical structure in the brain. So it seems to follow roughly an inverse trajectory to the way in which the sensory system is organized, going from simple features of the environment to more complex features, and then eventually to objects, faces, and things like that. It almost looks as though the artists have started right smack in the middle of the brain, which is probably because that's the area that we consciously have more access to, and then moved more peripherally over the course of history. So as we go as I go through the talk, you'll see that the actual things that my version of the history of art suggests is that we're actually moving more and more to stimuli, to visual presentations, representations, which activate the earlier parts of, say, the visual system. Okay, so that's where we're going. As I say, it looks as though we all now are phrenologists. These are two phrenological pictures of what the phrenologists believe. They started by, this is now back in the early 19th century, they started by believing that the brain was modular. Their mistake, I think that was correct, their mistake was they thought you could actually tell which parts of the brain were doing which things by studying the size of the changes in the skull. So people who were very smart had very, very strong frontal lobes, frontal prominences, and so on and so forth. They came up with a very strange category of psychological things to be mapped onto the brain, and I actually, I'm not sure you can see it here, but some of them are things like the first one, amativeness. So I started life as a classic scholar, so I know what amativeness means. And amo means to love. So this is the area of the brain which is actually involved in love. And I suppose that was a code name for sexual activity. But it's located in the wrong place. And then there's philoprogenitiveness, and so on and so forth. So you can see that you're dealing with people who are a little bit removed from the day-to-day activities of most of their fellows. Philoprogenitive is love of your children, and so on and so forth. And there are very, very strange things here, which I'll read from these things here. Secretiveness and contrariness and things like that. I think they got the categories wrong. But the basic idea is actually quite a good one. It does look as though the brain is modular. And in the early parts of the 19th century, late parts of the 19th century, early 20th century, people like Brodmann actually looked at the brain and divided it up into lots and lots of parts on the basis really of histology, just saying this part looks a little bit different from that. And over the years, we now know on the basis of lots of studies, I'll touch on a few of them, but really won't go into it in a lot of depth, that yes, we can actually parcellate, particularly the neocortex, into a whole bunch of subdivisions, and those subdivisions, we can assign, at least broadly, functions to them. So, as shown here, we can say that the areas in the back here in the occipital cortex are visual. We can actually say that the areas here are involved in somatosensation, audition, and so on and so forth. Some of these areas are really quite high level in this hierarchical structure, so we're still not sure exactly what they are. So The way in which it looks as though the brain is organized is, for example, if we look at the visual system, is that sensory information comes into the back of the brain, the occipital cortex, and then it goes through a series of processing modules, and each module kind of takes the information coming from a previous one and makes it more complex. It's easier to see it on, this is what a wiring diagram of the system looks like. You come in with from information from the retina here. You go through several way stations, get to the cortex, and then you go through this incredibly complex wiring diagram where you can, if you want to, you can see a kind of a hierarchical structure. You go from areas such as V1 to V4, and then to higher ones like that. And then eventually you get to areas that I'll talk about very soon, the hippocampus and the amygdala, which seem to be involved in really high-level processing of information coming from the outside world. And we know from a lot of work done using fMRI, work using electrodes in animals, and so on and so forth, that some of the things that the visual system is pulling out of this sensory array are things like the shape of objects. There are special areas devoted to place recognition. In fact, there are several of them. I just have one of them here. There are areas which are involved in recognizing landmarks, which might be used to construct our representation of space. Okay. And the way it works is that if you look at the very earliest part of the visual system, the way it works as though it looks as though you take several of the modules at one level and you put them together to make the representation at the next level. It's a very gross oversimplification, and it's clear that there's a lot of crosstalk between the modules, and it's also clear that there are feedbacks from higher levels to lower levels. But essentially, if you look at, for example, the way in which you take cells from the thalamus and you put their firing fields together, which are all roughly circular, and put them, the right ones together, then you get a representation of a line. And as Yuval Weasel showed a long time ago, the cells in the V1, in the early part of the visual system, actually only respond when a line is presented in the right orientation. So oriented lines. And you can see how that you could then generalize this. You could then go to the next stage where you put several lines together and so on and so forth. This approach was characterized as the grandmother cell approach, because of course if you keep going farther and farther into the system, you would effectively expect to see cells which represented your grandmother. And I remember back in the 1970s when I first started looking at a part of the brain I'll talk a bit about, the amygdala, I started finding cells which, for all intents and purposes, looked like grandmother cells. And we were still at the period when it was not really accepted in neuroscience to talk about grandmother cells with highly specific representations. And I remember Charlie Gross, who was the first one to find face cells and hand cells. He came into the lab and he said, well, what do you think about this? I said, oh, no, we're going to be in trouble. We're going to be, because most people still, even at that late date, thought that the brain consisted of neural networks which all talk to each other and where you wouldn't be able to find such highly specific cells. So I said to him, what are you going to do? He said, I'm going to bury it. And that's exactly what he did. The first paper that describes the face cells and hand cells is actually, there's a long, long paper, and then it's only at the end he said, oh, by the way, there are a lot of cells that respond to hands and faces. And I did the same thing. It's now, though, widely accepted that's not the whole story, but that's at least part of the way in which the system works. We still have a lot of work to do, but that's how it works. Okay, I want to start then by telling you a little bit about how the areas which are very deep in the brain, very far away from the periphery, how we think they work. And when we first started looking at these areas, a lot of people said, this is crazy. This is nuts. What you really need to do is you start here, and then you figure out how these cells work, and then you find out how these cells work, and you figure out how these cells work, and so on and so forth. If you're going to do that, it's going to take a lifetime. And I just thought, I'm not going to wait around for a lifetime to sort of find out what the areas, the cells in the areas that I was really interested in, these areas deep in the brain which are involved in representing abstract things like places and emotions and things like that. I thought, okay, I'm going to plonk it right straight into the middle here, which is what I did. And it turns out that, in a funny sense, the cells deep in the brain may be easier to understand in terms of what they're coding for, partly because they're closer, I think, to our conscious representation, our folk psychological representation of what's going on in our mind and other people's minds. And it may be easier for us to understand those things than things that are closer to the periphery, for example, in the retina, where the cells, the actual rods and cones are interacting with electrons. Who the hell knows what an electron is? Nobody knows what it is, right? We can write equations, but we really don't have a very good grasp of what it is. So to understand those cells is going to be very, very difficult. Anyway, let me tell you a little bit about what these two areas, the hippocampus and the amygdala, represent, and then I'll work my way back through the visual system, going in the wrong direction, and see if we can make some sense of it. Okay, so let's start with the hippocampus. Hippocampus is right at the end of higher order visual and other sensory processing. If you follow these, if you go backwards from the hippocampus here into the neocortex, you find that there are highly analyzed visual features that are sending information into it, and things like that. And those cells get their information from more simple cells and so on and so forth. Well, what does it do? Well, we're pretty sure that in animals and humans, one of its functions is to actually provide a representation of the environment. So right now, I'm looking out at all of you, and my hippocampus is going, wow, look at this environment. And look at all the structure here, and where are you? And I'm thinking, I hope I'm not going to be turfed out at the end of this talk and have to find my way back this complex. Okay, so we believe that on the basis of animal work, and we believe it on the basis of cells that we recorded in the animals. I'll show you in a second. But for a long time, we couldn't study this in actual humans. We couldn't study it because there was no way of looking at brain activity, and I'm talking now about the 1990s, looking at brain activity when somebody is wandering around a big environment, a building like this, or wandering around Christiana. So one of the things that we did, and by we I meant Neil Burgess, in my lab, was to actually take a video game, a first-person shoot-'em-up video game, and take it apart, removing all of the guns, removing all the monsters, removing all the politically incorrect aspects of it, so that all we were left with was a big, big environment, 70 by 70 meters, which is very complex, and which people would take about an hour or two to find their way around and explore and say, yeah, I understand how to get from one place to another. And getting from one place to another meant, if you were here, how would you get over here? And there were lots and lots of ways in which you could do that. Okay, so it's a complex environment. And it was just what we wanted, to look at how the brain actually represents of complex environments. So we asked people in the scanner, find your way from one part to another part. And here are some screenshots from it. The video game is based on a derelict part of Los Angeles. You can't see it here, but there's an old trash can which is burning. You can see this, the marquee is a little bit in disrepair. There's a very nice blinking eye here, very abstract eye blinking. I won't tell you what the original video was. And there's something for, if you want to play pool, you can play pool. If you want to have a drink, you can have a drink. There are lots of things. Okay. When we imaged people, what we found was that this area in the brain, the hippocampus, was actually activated by that part of the brain. So, and only one other area of the brain. And if people took good routes, if we asked them to go from A to B, then they went straight, and sometimes they went slightly deviously through this path, and sometimes they went completely wrong. And it activated the hippocampus as a proportion for how well they actually navigated. It's pretty good evidence that this part of the brain has something to do with representing the environment and things like how to get from one place to another. And then Eleanor McGuire came along and said, well, if there are these there are these really good navigators in the world. There are London taxicab drivers. So why don't we just look at their brain and see whether their brains change as a function of how long they've been actually driving. And so she showed, by just using fMRI, that the brains of really expert cab drivers were bigger than ones who were less expert. And the longer you were a cab driver, the bigger your brain got. So it turns out the phrenologists really were right. And there now is a whole industry of trying to show that people in certain professions have bigger bits of their brain than people in other professions. Okay, and there are cells in the hippocampus of rats which look like they are doing the job that you would expect on the basis of that work. They fire when the animal goes to one place and not to other places, and then another cell will fire in another place. So they provide information which you could roughly call a cognitive map. Okay, now that's not all the hippocampus does in humans. It seems to take that basically spatial mapping system, and it adds to it a lot of other things, including something about time, and it represents people's experience. So as you're going around the world, the hippocampus is actually logging your experiences and providing information about what you did in a certain place in a certain time. And the reason we know that is because of studies on this chap HM, which were originally by Brendan Milner and have carried on by Sue Corkin, who unfortunately died very recently, and who after a long time, after doing a lot of experiments on HM and other patients like that, concluded that what's wrong with HM is he doesn't have what we call episodic memory. He doesn't have the ability to remember what happened to him in the past and things that he personally experienced. And so she said, he cannot recall anything that relies on personal experience, such as a specific gift his father had given him. He retained only the gist of personally experienced events, plain facts, but no recollection of specific episodes. So this part of the brain then seems to take information in and compose it into a map-like structure of the environment. The other part of the brain, the amygdala, is generally thought to be involved in negative emotions, and I'll show you some evidence for that. But I think it's much broader than that. I think it tells you about the salient aspects in your environment. So the best studied patient with amygdala damage is SM, and she seems not to be able to understand the emotion of fear and surprise. And it seems to be that these two emotions are related in her world. So if she's asked to draw various pictures of people with representing various emotions, she can draw somebody who's happy, sad, surprised, although it's not terribly good, disgusted, angry, and then when she's asked to do afraid, she struggles, and she just can't draw something which is a picture of somebody who's afraid. And if she's asked to sort of look at pictures and characterize them and say, look, what's this picture of? Is this somebody happy, sad, and so on and so forth? If you look at controls, they can do this very well, but if you look at SM, she can do it all right if the pictures are showing people happy, disgusted, or sad, but she can't do it if they're showing surprise or fear. So this part of the brain looks like it's taking information in from those visual systems, the earlier visual systems, and actually saying, this is something which I should be happy about, sad about, and so on and so forth. It seems to be picking out the things in the environment which are of ethological relevance, of salience. Interestingly enough, SM, and this is something that I found very interesting, and I'll just say it as a kind of a side note. She also has trouble identifying people's their approachability and their trustworthiness on the basis of their faces. So you should all be sitting there saying, what are you talking about? And it turns out that if you show, usually American undergraduates, 50 or 100 pictures of faces, they reliably can say, yeah, I trust this one, or I wouldn't approach this one. So there's something about the actual faces that people are responding to. And people like Liz Phelps have gone on to show that this part of the brain is also involved in racism, This is the part of the brain which gets activated when people feel strong negative feelings about people of other races. So if you give people these pictures and you say, okay, which are the most positive ones, then controls are fine. And so is SM, except that she can't pick out the negative faces. She just doesn't say that people who would normally be characterized as unapproachable or untrustworthy are that. Okay, so a little bit more on the amygdala. There are cells, people are recording from the amygdala of patients who are undergoing epilepsy surgery, and so we know a little bit about the activity of these cells in humans, a lot more in animals. And there are cells which respond to different emotions. If you show people faces of lots of different stimuli, the cells will respond, some to anger, some to sadness, some to surprise. Surprisingly, one of the studies that was done in California shows that some of these cells in the amygdala are actually interested in animals. So they're taking the information that this is an animal and they're responding to that. Okay, and as I said to you already, work in lots of laboratories has identified lots of areas earlier in the visual system as actually doing signal processing, doing work on the various visual cues coming in, which identify things like the shape of an object, a face, places. Okay, right, so that's the neuroscience bit. Now you know everything you need to know about all the bit that I'm going to tell you on the second part. Because what I'm going to try to do is to actually say that different representational artists and different styles of painting have actually tried to address these different parts of the brain. Okay, so that's the story. And I'm going to go through the three roughly historically. So I'm going to start with the Lascaux cave paintings, the earliest representation of a figural representation that we have. These are paintings that were done 17,000 years ago in caves in the southwest of France. And you can see here, this is a picture of what the caves look like. They've actually no longer accessible. You have to, they've produced caves and paintings which are replicas because the caves were getting destroyed. If you look at what's actually represented on the wall, this is in the Paleolithic area time, you can see that it's mostly pictures of animals. And animals that would have been very important to the people who made those representations. There are animals here looks like an animal which is actually just about to do some damage to this chap here. There are people who are hunting animals. There are animals which look like they're hunting each other, and so on and so forth. Most of the representations are of animals. Exactly the sort of important stimulus that we think would be activating the amygdala. It'll activate other things as well. It'll activate some of the areas in the earlier neocortex, but those are the kind of stimuli that we would think would be actually activating the amygdala. Of course, the other thing that we would expect to activate the amygdala are pictures of people who are showing strong emotions. And here's something from Munch. And it's called The Scream of Nature. I'm sure you've all seen it. And again, we think this is really actually almost designed to activate the amygdala. What's interesting, of course, is it's not a very well painted. The actual face is not painted with a lot of precision. And I've always wondered about that. It's a kind of vague around the face, around the mouth. And one clue as to why that might be comes from work by Margaret Livingston, who was working in David Hubel's lab, and has written a very nice book on art and neuroscience. Because she was interested in the Mona Lisa, and she said, why is the Mona Lisa such a strong image? Why does it captivate people? Why are they so impressed by its ambiguity? And she did something very clever. She took the Mona Lisa picture and she filtered it with different filters. So she filtered it with, you can't see this here, with high-frequency filters, which is essentially taking the part of the object which is most accessible to central vision, where you have the finest grain discrimination. And she also filtered it in other ways, and essentially using low-frequency filters, which would actually mimic what things look like to the peripheral vision. And of course, when you look at a face like this, what you do is you look back and forth from the various parts of the face. And what you pointed out is that if you look at the smile, this enigmatic smile, it really looks as though, when you look at the high-frequency, very, very well-defined version, you see a bit of a smile, a hint of a smile. But then, as you look at the filtered version, you see that the low-frequency filters, the things you would see out of the corner of your eye, actually has a much broader smile. Very much, so there's a suggestion that maybe it's different parts of the visual information which is actually going to different parts of the brain and actually calling out different things. And so her idea is, as you look at the Mona Lisa, when you look away to her eye here, you see her smiling, but then when you look back to her mouth, you bring in the central vision, and she stops smiling. So the enigmatic aspect of the painting is because as you move your eyes around, you get a different view of what her emotions are. Again, this part probably activating the amygdala, this part activating more areas earlier. Okay, now in terms of the hippocampal representation, that of course is, as I say, it's representing large-scale scenes, things you can walk around in. And of course, for many, many centuries, really, artistic endeavors were focused on trying to sort of reproduce what it is in the image, which enables you to see this as a three-dimensional scene. And it was really only in the Renaissance when people like Piero della Francesca actually began to understand how to use projective geometry to be able to capture what it is about the 2D representation that will make you see it as a three-dimensional scene. So you can see here that here's a picture. This is the flagellation of Christ by Piero della Francesca, and you can see that it's got a lot of things. This is not the kind of stimulus that you want to activate to use to activate specific parts of the brain. This is going to activate the whole brain, and rightly so. So it's a emotionally strong stimulus. It's Christ being flagellated, and Pontius Pilate sitting on the corner here, and then these three Tuscan noblemen discussing the weather and the price of corn and why would Britain possibly leave the European Union. But what it captures is that by using projective geometry and getting all the lines to converge at some vanishing point, you actually get this very strong feeling of a three-dimensional representation. So that part of it, we think, would activate the hippocampus. But of course, as I say, there are lots and lots of other things. So it's not an ideal stimulus because it'll activate lots and lots of parts of the brain. To start getting down to things which will activate more specifically, and that's of course what you want to do, I suppose, is to activate just one part of the brain and do it as best you can, and that's something you're going to really find beautiful. You need to start looking at things like just pictures of landmarks, pictures of buildings. And of course, if you, and the other thing that people started to think about was, well, a picture such as the one we just saw really doesn't capture what's happening in my early visual system. What's happening as I'm looking around is I'm getting a little snapshot of this, a snapshot of that, snapshot of that. Whereas a lot of the pictures up until the 19th century seem to capture some eternal, constantly available, holistic type of image. So what people like Claude Monet did was he started saying, I want to actually try to capture just the impression that I get from a single snapshot. And so he would do things like take something like the Rouen cathedrals, and he would do lots and lots of separate paintings of it, trying to capture the way in which it looked at very, very specific instances. And of course, since it's a building, then it's going to activate the place representation here. And interestingly enough, you can see that, for example, the color changes, and these series are usually taken at different times of the day and under different lighting conditions and things like that. So lots and lots of things are changing about this, and that's what he was trying to capture. And he said, the older I become, the more I realize of that I have to work very hard to reproduce what I search for, the instantaneous. He's trying to get what the world looks like from a snapshot. The influence of the atmosphere on the things and the light is scattering throughout. Now notice that these buildings can actually are the same representation, the picture's the same representation, the same building, despite the fact that the color is changing tremendously from one to the other. And that's, of course, because you don't actually see the shape of something on the basis of its color. Those two things are handled separately in the brain, as far as we can tell. There's a shape area, and that's different from the color area. And that's, of course, a big, big problem, as I'll point out in a second, is given that these things are separate in the brain, how do you put them together? Is there an area that puts them together, or how do you put them together? Well, this idea that you actually can represent things, the shape of things, independently of their color, then took hold with the Fauvis and also with the early Henri Matisse. And it brings up this binding problem. How do you know that a color that you see goes with a particular shape? And of course, the way they explore this is to actually deliberately give objects, familiar objects, very, very contrary colors. So instead of the river Thames here being green, its usual green, the sky blue, and so on and so forth, they just painted things which aren't quite arbitrary. They're not arbitrary, but they're not the naturalistic colors. And yet, once you get used to it, you look at this and you say, yeah, I like that, it's fine. It's activating some complex of activity in my brain, and it doesn't bother. And the same with Matisse. Matisse would take a figure like this, the femme au chapeau, and he'd say, I don't really need to use the color to actually identify this person. I'll use the color as a means of expressing my emotion. And that's because the luminance of the color, the actual relative brightnesses are being reflected on what we're using, one part of our brain is using to identify the object. Well, once we start thinking about color, then of course, people started saying, well, maybe we can actually represent color directly and begin to explore the way in which color is created and the way in which colors interact. And I remember when I was doing philosophy, a philosopher said to me, he said, this color is really mysterious. It's really just a series of wavelengths, and yet, instead of a continuous variable, going from one end of the spectrum to the other, I see discrete colors. And of course, the problem was that he was wrong about how you see colors. You don't see colors by just looking at wavelengths. You look at the relationship between wavelengths, and it's the fact that there's a yellow here and a white here and a red here that actually gives the color to it, because the brain and the color areas, particularly in V4, are actually using comparison between the reflected wavelengths, different wavelengths. And Semir-Zeki, who actually discovered V4 and discovered the color cells in V4, actually uses Mondrian's. He uses that as a stimulus. It's not a terrifically analytical stimulus, but as you can see, it's not quite clear exactly which parts of the painting that the cells are responding to, but it's actually it's very interesting. Other things that people have tried to start pulling apart these stimuli in the world and seeing if they relate, how much you can pull them apart. Or, for example, if we think about motion and how a succession of movements is perceived as a continuous movement, and what would happen if you broke that succession up into its individual parts. And of course, this comes out of the work by Muybridge and Jean-Étienne Marais, who in the late 19th century used stroboscopic flashes of lights or actually put lights on subjects and began to see, as it's shown here in this thing from Mare, this picture from Mare, that you can actually describe somebody's motion just by the movements of the lights that are out. And this is now used in psychology quite a bit. And it was the inspiration for Duchamp's new descending a staircase. It's as though time didn't exist, so that each successive representation of the movement is actually contained all in one. And Dave Perrett has now shown that there are cells in the infratemporal cortex which respond specifically to movement of animals and humans. So that all you need to do is put lights on someone and the cells will respond to that very abstract thing of movement in space. And finally, once you're starting to pull things apart, you can say, okay, well, there are areas in the brain which are actually representing faces. But how do they do that? Are they doing it by holistically? Are they doing it by having the whole face and responding to that? Or are some of the cells actually taking bits of the face? And so people like Freiwald have actually used complex stimuli where they've taken faces and cartooned them and then broken them up and said, well, here's a cell which responds to the face and also to a cartoon of the face, but if I give it just the eyes and I give it the mouth, will it respond to that as well? And you get all sorts of combinations of components. Some of the cells only respond to the entire face, but some of them respond to combinations. And you can see how the cubists, and you can see how and people like Baccioni could start to think about, well, can we think about faces as being pulled apart? Can we activate those cells which respond to only components of the face rather than to the whole face? Okay, and I'll just mention Bridget Riley and her fantastic stimuli which look like they activate the motion-sensitive parts in the brain. So we would think that that kind of a stimulus, which looks like it's moving all the time, would actually activate MT and areas that are sensitive to movement. And then finally, but not quite last, but close to last, right coming up to the present, if you look at what the abstract expressionists were doing, they were breaking the whole canvas down into the simplest configuration. So Barnett Newman quite often will have just a single stripe against a black background, or Rothko will have one or two actual bars against a field. And that looks like they're getting at the earliest stages of the visual system. It looks like those are stimuli which are actually driving the cells in V1 or in the earliest stages. Almost as though that we've progressed from highly complex emotional-like stimuli, complicated representations of the environment, to working our way all the ways out, now in abstract expressionism, to very, very stimuli which probably are activating solely the earliest parts of the visual system. And finally, what am I going to do with Jackson Pollock? I don't know what that's supposed to activate. I cannot believe there's a part of the brain which is going to respond to a Jackson Pollock. So, question mark. The theory doesn't cover all of the data. There's room for more experimentation, and we need another grant. Thank you very much.